Thanks a lot for your very nice introduction. So I will be talking about oaks in the upper Midwest today. And we heard about oaks hybridizing quite frequently. So there is apparently a lot of interspecific gene flow where species co-occur, but they are still distinct. And we ask ourselves, how is this possible? There is a lot of gene flow. Why are there so many species in different environments? And my talk tries to address this question partly. So I will give a general introduction about oaks, and you know a lot about oaks, so I will not talk too much about this. I will provide some evidence that there is actually gene flow among many species within sections. Then I will look at specific species pairs and look at the overall genetic and morphological differentiation of these species. And I will look at non-genic randomly selected markers, but also at markers that are located within or close to genes. So we are looking in principle at specific regions in the genome that show actually these signatures of speciation, that show that they are actually distinct. So in an earlier talk, we've seen like if you randomly select some markers, we might not see any structure actually, but if we look at a genome-wide scan, we might be able to distinguish these different species or whatever we want to call it. So my interest is in identifying these regions and also to see is there any association of these gene regions with traits that actually that distinguish between species. So there is evidence from artificial crosses actually. So there is many of the oak species within sections and even across continents, they are crossable if you force them to, but also in natural population, if you perform like gene flow analysis within the stands, you can actually see that there is also gene flow in natural population. And that's an example of a European oak here in the middle. That's sessile oak, Quercus petrea. And we collected seeds from this tree, and we identified like the genotypes of all the individuals in the stand. And actually we've seen there is a lot of gene flow from outside the stand, but the ones that are identified as pollen parents, they are actually not only the same species, but they are partly interspecific hybrids as identified by genetic assignment analysis, and they are partly different species. So apparently there is gene flow in the seedling generation that is evidenced by paternity analysis, but if we look at the distribution of the individuals in the stand, we see they are not randomly distributed, so the species are clustered. So they are not randomly admixed. And if you look up in the upper Midwest where I live now, actually we have like virtually only Quercus rubra there. But then our forester showed us some little shrubby trees down on these outwash sands. And they are just smaller, but they're very close to these Quercus rubra stands, and we didn't know exactly is this the same species or is this just phenotypic plasticity in a different environment. And in Europe we see a similar pattern, so there are species that are kind of stratified along soil moisture gradients, and a very similar pattern we see in the upper Midwest, on the upper peninsula actually, for Quercus rubra, and for Quercus, what we think might be Quercus ellipsoidalis. And I will provide some evidence genetically that we actually see these are ellipsoidalis individuals. So we collected also seeds from these neighboring populations of northern red oak and northern pin oak, and actually it appears they are not only morphologically different in different environments. If we plant them actually randomized, here they are sorted by species, but earlier they were randomized, we see there are pronounced differences in even aged seedlings in size, actually. So there were really pronounced differences. So these northern pin oaks are very little, and this is here a Quercus rubra, the same age from these two neighboring stands just a few kilometers apart. So there appear to be also genetic differences between these two populations, which would indicate there are two different species, actually. So what we are looking at, and that's a very simplified representation of the genome in oaks, actually. And when we performed, or others performed, like genome scans, looking at many genetic markers. So actually, as a result of interspecific gene flow, many of these markers that are here, like listed on a chromosome, they show very low interspecific differentiation. And this is a measure of interspecific differentiation. But apparently, there are a few markers that show surprisingly high interspecific differentiation that somehow resist the homogenizing forces of interspecific gene flow. And we see different patterns in poplar, for example. So we looked at the interspecific differentiation using also microsatellite markers. And it was already mentioned, so these are multi-allelic markers. They have many different variants within a population, and they are mainly located in non-genic regions, but they are also located in expressed genes. And we took advantage, actually, on these European projects that developed a set of gene-based microsatellite markers that are here located on these chromosomes relative to each other. And most of these markers are actually gene-based microsatellite markers. So these have the advantage that they are relatively inexpensive, but that you can also look at genetic variation within or close to genes. So we used these resources actually and applied them, these markers that had been developed in European white oaks, to North American red oak species. So, and when I arrived first on the Upper Peninsula of Michigan, so I looked at some of these stands, and actually we wanted to collect some rubra and ellipsoidalis, and this is in the Ural Mountain Reserve, close to Lake Superior. So there are actually like Quercus rubra plants that look very tiny, and they have a very wide variation in leaf morphology that is actually reminiscent of like other species, but it might be also a reflection of the different environments. So we developed a set of these microsatellite markers, mainly transferred them from European white oaks, and we looked in an initial experiment at like interspecific differentiation patterns between 4 relatively closely related oak species of section Lobate, and we see here the distribution ranges of these species. They are morphologically distinct, but they show a relatively wide morphological variation. So, and they also show differences actually in their requirement for soil moisture, and they are kind of listed here by decreasing water requirements. So we collected samples from each of these species, and the question was, is there actually gene flow between them, and are there certain regions that do not show this pattern of interspecific gene flow, that are potentially outliers involved in adaptive species differences with regard to water requirement? So we selected like mainly from the Midwest. So these are all these population pairs of Velutina, Quercus coccinea, ellipsoidalis, and rubra. And then they have been identified morphologically. And when we looked at them at this set of gene-based markers, we found actually there are 4 genetic distinct groups corresponding to the morphological species. But as you see, not completely. Each of these columns here shows individual samples belonging to one of the 4 genetic clusters. And you see this looks good for many of them, but there are a certain amount of individuals that are genetically intermediate, in that case, mainly between Quercus ellipsoidalis and Quercus velutina, and their ranges are overlapping. So that's an indication of interspecific gene flow. It might also indicate that they are just recently diverged and that they share certain polymorphism. But interestingly, when we excluded the hybrids, actually Coccinia is a sister species to ellipsoidalis, and their ranges are parapatric. They are barely or not at all overlapping, and we see actually there is not an evidence of introgression here between Quercus coccinea and ellipsoidalis. So I want to provide you some evidence for interspecific gene flow, and I don't want to show all of this, but we also did paternity analysis, and we looked at chloroplast markers, and these maternally inherited chloroplast markers, they were actually also shared among species, and like regionally restricted chloroplast markers were shared among neighboring populations, which strongly indicated that there is interspecific gene flow between most of these species pairs, between ellipsoidalis and velutina, and between ellipsoidalis and rubra, and most of the other pairs. We see that the gene flow actually is not completely symmetrical. So these are the ellipsoidalis, morphologically identified ellipsoidalis individuals, and hybrids and introgressive forms between velutina and ellipsoidalis are mainly found in the ellipsoidalis. So there is evidence of asymmetric gene flow, but you see, like for the different regions, the level of interspecific gene flow and their direction varies, indicating that there might be some effect like of flowering time or of environmental conditions in general having effect on the patterns of interspecific gene flow in these species. So then we looked at a set of these markers, and we just compared like the level of variation with the FST, the interspecific differentiation in percent, and we see that most of the markers, when we looked at them, they show an interspecific differentiation of less than 10%. So most of the alleles are actually shared among species, but we find exactly this pattern that a few markers, and these are mainly gene-based markers, they show a much higher differentiation, indicating there's potentially selection. So that was an initial study that we performed, but then we focused actually on the upper Midwest, where I live now, on the two species that show the most pronounced differences in drought tolerance, Quercus rubra and Quercus ellipsoidalis. And we also try to collect samples in contact zones between both of the species. So our sampling is mainly here in the northern distribution range of both species, and actually there only these two species occur. There is no confounding effect of hybridization with other closely related species. So these are the sampling locations, and the question that we ask, are there actually certain genomic regions that are differentiated between species pairs that occur in contrasting environments that are found repeatedly across these different population pairs? Are there certain genomic regions that are really describing species differences and that are potentially, what we ask later, that are potentially associated with traits involved in these species differences? So we did these replicated selections and collections, and actually ellipsoidalis is always like on a drier soil, mainly on sandy outwash plants. So we have them all in gene flow distance, but on contrasting soils. So then we used genetic markers, again these microsatellite markers, to see whether they are morphologically distinct in a way, but we confirmed that with actually these genetic markers, And we see again like there are two genetic clusters this time, and the two genetic clusters correspond to the species Quercus rubra and Quercus ellipsoidalis. We again see there are potential interspecific hybrids and like misidentified individuals. So there is again evidence for gene flow and hybridization. So we selected only individuals that were clearly identified as species based on morphology and based on genetic markers. And then we screened a larger set actually of gene-based microsatellites, which are these EST SSRs and nuclear SSRs that are not located in genes. And when my student showed this, I was a little bit disappointed since this is not a nice figure. So I've seen that, and every point is like in your expectation, what you would expect under patterns of interspecific gene flow. There is no signature of selection here. And I said, this is a nice picture, but it doesn't show anything. There is no pattern of selection, but I didn't see this point here. So this is really, and I said, no, that must be a mistake. So the analysis is called outlier analysis. And I thought, yeah, that's an outlier, but that cannot be true. So please look at that, and like this point, I mean, you even oversee, you don't see it. So what is the genetic structure? What is the frequency distribution at this specific marker? Is this true? So what that would indicate, the differentiation between these two species where there is apparently gene flow between them, it's like nearly 80%. And it was like in the other population pairs, it was between 40 and 50%, but it's still very high. So that would indicate that there are actually two different variants, mainly in each species. And if you look at this in more detail, that's actually the case. And remember, that's a gene-based microsatellite marker. This is the size of the fragment, 138 base pair, 141, and these are the genotypes. So these are homozygous. They have 2 times this 141 allele. They have 2 times the 138 allele. These are the heterozygous. So they are virtually, the two species are nearly fixed on alternative alleles at this microsatellite locus. And the two alleles are exactly 3 base pairs apart, so they are 1 repeat motif different, and they are located in the gene. So that could be something of interest. So we looked at these species, at the distribution then in different generations, seed and seedling generation, and actually we looked also in the 3 other species, and we found like an interesting pattern, like again, like in Quercus rubra, it's mainly the 1 allele with 148 base pairs. And in all the 3 other species, like the other allele is predominant. So, and remember, there's evidence for gene flow between all of them, but this very strong frequency difference is somehow maintained in the population, and we think it's only possible by very strong divergent selection. So we started to look a little bit at the sequence of this, and we, in principle, sequenced the PCR product, and we confirmed that actually this polymorphism, so the indel of 3 base pairs, is actually located in the coding region of a gene, which is a constans-like gene with potential role in flowering time and growth in poplar. And we see like that the variation is due to like insertion or deletion of 1 amino acid, a glutamine residue. And we don't know what the function of this is, but interestingly, variation in this poly-Q repeat in poplar resulted in slower growth in certain varieties. And we know that this poly-Q repeat is important for like genetic disorders in humans, but there is not much known about this poly-Q repeat in plants, actually. But it's a very surprising result that actually a difference in the coding region actually shows like this distinct differences among species with different adaptations to drought. Interestingly, the group in Pierrotin actually looked at the same constant-like gene and looked at SNP variation in that gene, and the same gene was showed a weak but significant association with vegetative bud burst, which is an interesting result. So that led us to the question, how could we find such an interesting marker that has potentially a role in adaptive divergence between species? How can we associate variation in this gene with, for example, with flowering time or with differences in drought tolerance? And this is now speculation and what I'm hoping for that we can do in the future in cooperation with Andrew Hipp, so that we actually look at these red tag markers, that we perform genome scans and look at many, many gene markers at the pattern of interspecific differentiation and identify these interesting outliers as the one that we identified and actually map them back on a genetic linkage map that is hopefully anchored to a genome, which would be the case for Quercus robur, where the genome is sequenced. And again, this is speculation, and we hope to do this kind of thing. So we would like to combine these two sets of information, the outlier loci that show these high interspecific differentiation, like the constans-like gene, and we would like to include these markers on a genetic linkage map and to see is actually this genomic region, this chromosomal region, associated with traits of interest that actually distinguish these related oak species from each other. So we haven't done that in oaks, so I talk just 1 minute about poplar. So we have an interspecific poplar family, which is a backcross, an interspecific backcross, and there we looked at leaf morphology actually. We would like to do that in oaks too, but we did it in poplar, and I just show that since there we have a reference genome, and I should say we didn't do much of the work. Matthias Kirst in Florida did most of the work, but we found we have this progeny up there where I live now, and we found a very strong QTL for leaf shape variation. And I wrote an email to Matthias Kirst, and he found actually the same QTL in a different environment, nearly exactly the same position, and he also looked at gene expression patterns. So he found that actually underlying this QTL, there is a gene that is differentially expressed. So it was mapped as a phenotypic trait. So what I just want to show you here, if we have the reference genome, we can actually do that, combine these outlier screens that I earlier described, and try to see whether variation in the underlying genes, and there are still many, that variation in the underlying regions are associated with traits of interest that distinguish these species from each other. So that's in principle. I stop here, so that's just the outlook. So we would like to do in the future genome-wide outlier screening. We would like to anchor this to a linkage map and a reference genome and identify QTLs for the traits, but there is a lot of work and we have done very little of this until now. So we have to thank the funding organization, and actually I haven't done any of the work, so there are some of the students that did actually the work, and some of my dear colleagues on this picture. Thanks a lot.